It's my very special pleasure to introduce you today to Detlev Arendt. Most of you might remember him from yesterday when he was talking here in this very same auditorium on the evolution of bilateral brains. Okay, so for those of you who are not very familiar with Detlev's career, I will mention just a few things about that. Detlev studied in Freiburg, where he did also his PhD. Then he moved here to Heidelberg, where he did a rather short but insanely successful postdoc, to then move into running his own group in the developmental biology unit up at EMBL. And the motto during the leitmotif during Detlev's career has always been to try to understand the evolution of the animal body plan in general, but the central nervous system in particularly, and even more particularly the cells that form that central nervous system. And that started very early on Detlev's career, very, very early on. And I want to emphasize that just as a call for the many young students that are in the room today. Very easy to identify because you sit in the bottom half of the auditorium. So as early on, so during these very early stages of his career, Detlev has written a number of articles and letters that were highly influential and that also shape the future of his career. So back at that time, there was this question on whether, or there was the belief on whether the ventral nervous system that are in insects and the dorsal nervous system that are in vertebrates were have arose independently, or they had a common origin. And it was so these early studies are characterized by lots of reading and thinking and very, very careful observations on samples to then come to the very, very clear idea that they might have a common origin with a simply inversion of the dorsal ventral axis. And if there was a common origin where clearly the spectrum of on animals in which we could start studying that increased a lot. And that's what Detlev did since the very beginning, trying to find model organisms in which that question could be addressed. So I think that he constitutes one of the best examples on how, if you want new answers, you clearly need to look not at the very same models that everyone is looking at. And that model that they started working with when Detlev started here was platinaries, a model that back then was, most of us didn't know about. Now it's a very, very well-known model that you have heard some yesterday, and you will hear even more today. So during his career, Detlev has received funding from very prestigious agencies, has published very, very influential papers. He's member on many academies here in Europe. And again, it's a very special pleasure to have you here today, Detlev. We are very looking forward to your presentation.
Thank you. Thank you very much, Lazaro, for the kind introduction. It's indeed my pleasure to be here today and present on our research. It was also very nice yesterday, actually, with the pupils and also some of the students. And today's topic is in a way related to what we already discussed yesterday, but the approach is different. You will see there is at the end not that much overlap, but it is both touching, and Lazaro mentioned this already nicely, on the topic that has fascinated myself and then has been the topic of the lab for a long time now, which is the evolution of the brain. And it is intriguing to think that the evolution of the brain is actually, as an evolutionary problem, not solved. We don't know where the brain comes from. So we know it with invertebrates, it works. So we have some idea that how the brains between, for example, lamprey shark and our brain are related. They have the same structure of the brain. But beyond vertebrates, which was an evolutionary term from urbilaterians to vertebrates not a long time, we don't know much. So how the vertebrate brain came together in the beginning. The same on the insect side. Yeah? So we lose track once we leave the big phyla and think about how the brain looked like before, we have not much of a clue. And this directly touches on this image that I showed there, where you see animals that are molded after fossils that we know from the Cambrian explosion. We know that, and if you see them like this, I mean, it is kind of obvious that there was a brain already in place. These animals were moving, many of them swimming or creeping on the ground. And during this time, there was an explosion of body plans, obviously. Slightly before, there was the first bilateral living. And we are interested in the question, what did their brain look like? And what were the very origins of what we have in our body as central nervous system? And that question has carried us for a long way. We have followed different approaches to tackle that question. In recent times, we have started to compare nervous systems, entire bodies, at the level which is fundamental to these comparisons, because cells are the units of life. And if you think in terms of cell types, it becomes clear that to understand how animals relate, it is very important to compare them at the cell type level. Now, what are cell types? So I first explained you how we approach this. Cell types are cells such as rods and cones that you have in your retina, the different neuron types that you might be aware of. Yes, so what are cell types? And I will start. So basically, we usually define cell types phenotypically. Yeah, we have drawn some cell types here. You recognize the rods and the cones of the retina, for example. They are defined by their morphology. They have a specific shape, structure, molecular machinery, and connectivity. And all of this we can compare between animals. And we have, to refine the concept of cell types, we have invested a lot into defining what a cell type is at the genomic level. And here I would like to stress today that a cell type needs to have its own regulatory program. So to be a cell type in evolutionary terms, it needs to be genetically individuated, and this means that there must be information in the genome that is only used by those cells, by this cell type. And this is often a specific combination of transcription factors which form the so-called core regulatory complex, and only then mutations are possible that would affect only these cells. And you can then also reconstruct how one cell type turns into two by the evolution of sister cell types. And that means that there are two different set of flavors of one and the same cell type that then become distinct over time. And you can see this, for example, here we can assume that there has been a ciliary photoreceptor precursor before, and this cell type then evolved into two directions, like rods and cones in our retina. And this involved mutations that established two regulatory identities instead of one. And these then very often mutually repress each other. And this way we then have distinct regulatory programs that control a certain division of labor and specialization. And we can then imagine evolution as the getting more and more, as a diversification of cell types that get more and more distinct. Wonderful. Yes, thanks, Jan. And so we have certainly in our body close to 1,000 cell types, but the cell types we have in our body are exactly a product of diversifications like this. There is something like a cell type tree in our body. And if we want to understand, for example, the evolution of our eyes, we can compare these photoreceptor cell types to other cell types in other animals. And this is possible via the genetic comparison. And so we have molecular signatures, and in essence, so these are cells from our Platinereus. Lazaro mentioned already, that's our model organism. This is from a volume EM, electron microscopy volume. What you see here are segmented nuclei, so that means the computer has recognized the nuclei and has also segmented the chromatin. This is why you can see it in different colors here. And what is reflected here is that all of the cells in the body have a different chromatin. And this means not more, not less, that this here reflects a certain unique open chromatin pattern that every nucleus has. And I can tell you already that we have done this for our organism, and we can then ask the computer, if this is here a certain cell representing a cell type, and we have this open chromatin pattern, so what are the most related cells in terms of open chromatin pattern? And what we manage then is to recognize, for example, the direct bilateral homolog on the other side that has a similar open chromatin pattern with very high accuracy. So this is among the most similar cells that the computer recognizes in the body, just reflecting this open chromatin pattern. And this is exactly what I was introducing before, the specific gene regulatory programs that a certain cell types has, and which makes its identity, so its individuation. This is, in a way, an optical way to represent what I just explained you theoretically. So a cell type is a group of cells with its unique regulatory program that evolves on its own and for which you can drive trees, build trees. So what we do in the lab for some time now, and this is what I'm going to tell about today, is we have a zoo in the lab. So we have a lot of marine invertebrates mostly, also some vertebrates. In essence, we are interested in the question where our nervous system comes from. Yeah? This is from the Voyager, by the way, if you don't recognize it. All of these animals are not chosen by chance. What we are interested in is the ancestry of humans. So this is the ancestry of our great-grandfathers and so on. And at some point in this lineage, there was a gnatostorm ancestor, which was the last common ancestor of us and shark. Then there was an ancestor that was the ancestor of us and lampreys that live today. There was an urchordate, which was related to Amphioxus and us still. And the very important common ancestor is this one. So there was on the planet, maybe something like 540 million years ago, there was the so-called eubalaterian alive on planet Earth, which was an incredibly successful species because it gave rise to us and also to flies and to our earthworms, or to our annelids that we are working with, these Platinereis, to mollusks. And this is where we need to understand what the nervous system looked like, and what we can do to find out is to work on the nervous systems of these representatives and also of animals that lived before, that branched off before, such as cnidarians, and they are also investigated, as you know, here at COS. Yeah? And so for all of these animals, what we do is we dissolve them into cells. Yeah, I told you cells are the units of life. And then we use single-cell sequencing. And so what this means basically is we isolate and we capture the transcripts from individual cells and sequence them. And this is a way to probe the uniqueness of gene expression in all cells in the body. And what this then leads to is, so single-cell sequencing, is that you get not only the general genes that are expressed in every cell, but you also get the unique ones that are characteristic for each cell. And so I want you to understand what these are. I think most of you probably know, but these are so-called UMAPs of gene expression in gene expression space for these animals here. So to explain it a little, so every cell has a unique position in this space with regard to the genes it expresses. Yeah, for each of for example, 30,000 genes that you find in a shark, for each of these cells, there is a certain value of expression for each gene. And according to these values, every cell gets a dot, and the closer the dots are, the more similar the cells are in terms of gene expression. What you can already see on these colorful schemes here is that gene expression space is not homogeneously inhabited. It is actually quite distinct. So you see single clusters here. Yeah? So these are cells that are very similar in terms of gene expression. And here we are with the cell types. So these are certain cell types with cells that resemble each other in terms of gene expression. They are not identical, but they are very similar. And here you can see that with this new technological revolution since a decade or so, we can capture all the cells of an organism at once and then compare them. Why can we compare them all of a sudden? Because these genes for these genes, for each of them, we can establish phylogenies and probe which gene corresponds to what. So at the end, we can compare these genes and cell types in expression space across species. And this is what we are doing, and I will explain how this works. Yeah? With emphasis on the bilateral, so comparing gene expression in all of these. And I will start with the concept of coregulons. And to illustrate this, talk about this organism that we also investigate in the lab, a polyplacophorn, a chiton. I'm pretty sure you know what that is. It is a mollusk, yeah, that looks segmented, at least has several body parts, and has a shell, of course, as other mollusks. And Tim, when he was a postdoc in the lab, was starting the chiton work together with Nico. Both of them now have their independent positions in Vienna. And Jake was also part of this project. He is now, since last year, he is a professor at Yale. And polyplacophorns are mollusks. This is now what they look like as entire animals. The body size is below a centimeter. And this is a reconstruction of their nervous system from this independent publication from 2018. We like this nervous system because it is very simple. It is quite representative, actually, for other mollusk nervous systems as well. And with this lateral component and this ventral component, also, and some specific parts in the brain here that have all received different colors, it could also be actually quite representative for the nervous system that existed at the beginning of bilateria. I will come to this. Yeah, so this is a nice nervous system in that it might be, in its composition, it might be quite ancestral. And then we do what I just explained. We take the animal, we dissolve it into its cells, yeah? We do single cell sequencing. For each cell, we determine for all genes of the genome whether they are expressed or not and in which quantity. And then the cells are placed in expression space. And for example, these cells here are quite isolated, as you see. So they are very specific in what they express. And, but then similar to each other. Other clusters are bigger, like this 6.2 here. And all of this are actually some kind of sensory neurons. And so we can determine sensory neurons are here. Sorry, these are cells from another kind of neurons, actually. These are sensory neurons. So what this means is we can determine the characteristics of the cells and cell type for the entire body in one go. In a way, this is for comparative anatomists, this is really a revolution in comparative data that we can take and compare. And then you can, for a cluster like this one, 1.3 or 1.7, you can take the genes that are only expressed in these cells, do in situ, whole mount in situ for these, and then also determine what these cells are. So for this animal, we have done this with normal whole mount in situ hybridization, and then we can place these cells in the body. So this is, by technique-wise, pretty pedestrian. So we have or Tim, who has done all of this, has placed them in the ventral nerve cord, the motor neurons, the sensory neurons. So this cluster here are forming this lateral strand that I showed you before. So you can actually make sense of these clusters of cells and place cell types in the body. This is a so-called cell type atlas for an animal. And for the marine invertebrates, what is nice about these cell types atlases is that we do it for the entire body. This is complete, yeah? And this also means our comparisons are complete. We can really say, if we, for example, compare this to an annelid, we can say this cluster is really the most similar to the one in the annelid because we have checked the entire other body. Yeah, so this is all of a sudden a possibility to do comparisons that are really exhaustive. And what we do then, we define so-called coregulons with a technique that is called weighted gene co-expression network analysis. And this is in a way easy to understand. Just understand that these are all the cell types that I've just shown you. They are listed here. And then we have asked the computer about sets of genes in the genome that are showing the same covariation across cell types. And if there is a certain set of genes that has a shared expression profile across all cell types, this leads to a row like this, where you, for example, for the very first one, you see that these genes have a very specific characteristic and that they're expressed in this cell type and almost nowhere else, or basically nowhere else. So, and other genes, like these ones here of this group, they have maybe expression in 3 cell types and then nowhere else. So they are quite specific. And what you can also see is that in the way, it illustrates again what I showed you already. Gene expression within, if you compare it and relate it to the entire body, is quite specific. Yeah? Just to illustrate this a little more. This here is, for example, representing the midgut. These 3 cell types together are the midgut. And then you have this group of genes representing this coregulon is only expressed in this cell type. And then you can ask, okay, which genes are these? And what you get then is a whole list of genes that show the same pattern to be expressed in this cell type, maybe a little where else, but mostly there. And this is clearly due to some common regulation of these genes. It's a regulatory program. Yeah? And that means all of this, what you see here, indicates different regulatory programs that exist in the mollusk body. So in the mollusk genome is not expressed everywhere, but there are many genes that are expressed in specific cell types only, and they show common pattern. Yeah, so this I think is important to understand. And in the midgut, we have, for example, this coregulon, but also this one, where you have genes that are expressed in all midgut cell types. It's this one. You see they all have, these are now the individual genes. This is the way to represent them, and they are all expressed in the 3 midgut cell types, and it's a long list that show the exact same pattern. And if you look what these genes are, they are digestive enzymes, iron and cholesterol transporter, and so on. And so this is a very specific molecular signature for the gut in the chiton. And this is information that is extremely valuable for the comparison. Yeah? And it's clear that these coregulons need to be regulated somehow. And this happens with transcription factors. You know transcription factors, they turn on genes, very often in combinations. And if you then ask the computer, okay, what are the transcription factors that are specifically expressed in these cell types here, you get these transcription factors. So this is a transcription factor list. And what happens then is that you get a short list like PTF1A is there, HNF4, GATA456. And let's say for developmental biologists who know their transcription factors and know what is usually making a gut, these are old friends. So we know these factors from vertebrates, from Drosophila, even from sponges actually, where there are these choanocytes. They are always making gut, and in a way this coregulon then is something that is very old and always existed in animals. Then we can do our comparison and conclude that something like a gut-like coregulon existed in the last common ancestor of all these animals. This so much about midgut. Then there are also the sensory neurons in the body plan and also motor neurons in the chiton that I already showed you. And for these, we also get a list of transcription factors that is illustrated here. Here are, for example, the STEADs. These are well-known sensory cells of the chiton. And also for these, we get transcription factor signatures like these ones here. And what I would also like to show you is that a trait like this shell in the mollusk, so the calcified shell that is very specific for mollusks, you can then ask what coregulants do they use, and then one of the coregulants that is in the shell field as well as in other cells is this one here, and it involves shell spicules, visceral cardiac muscle, secretory cells. It's a very specific pattern that these genes show in common. It's also a long list. And then if you look what this is, these are all genes involved in glycosylation. So this is a module, a genetic module of cells that secrete or that have a specific set of enzymes that produces that produces sugar chains that you find on proteins. So this is a very specific characteristic of these cells, so you can make sense of these modules. Good. And when you then do this for many animals and for many species, for all of their genes, you get something like this. So we are looking for conserved spiralian cell type families. And what we have done here is that we compare coagulons pairwise between species, and these are all different spiralians. And we calculate for these sets of genes pairwise distances, and this way then get conserved cell types families that existed in the last common ancestor of all these animals. So this illustrates how in principle you can go about finding conserved cell type families across species. And the sensory neurons and motor neurons are these two here, and what is important to see is that these are quite distinct. So there seem to be two fundamentally different cell type families conserved in these animals. Now some data on our beloved model organism, Platinureus dumerilii. This is the main animal in the lab that we are working with. Platinureus dumerilii lives in tubes. In our lab, it also builds its own tube. So these are self-spun, it produces them itself. It's a mesoherbivore, so it eats pretty much everything, but mostly macroalgae that it lives on. And this is a very nice model in that it takes a few months only to close the cycle and have a new generation. So it lives in labs since the 1950s of the last century. And what the stage we are mostly focusing on is this one. And you can see that with an antibody staining against acetylated tubulin, so showing you axons, this has an elaborate nervous system already, so it's an ideal animal for nervous system comparisons. So this is the stage I'm talking about. This is a young worm, so-called young worm. It is 6 days old. It has eyes here. It has the sensory organs of the head. It has 3 pairs of so-called lateral appendages with chaetae that it uses for some primitive form of crawling. It also has cilia with which it can swim. You see some of these cilia here. And this is, meanwhile, a very well investigated animal. It's an animal model for which we have made many contributions, but other laboratories as well, and I'll come to this. For this animal, for example, for this stage, we know that it contains about 12,000 cells. What we have done for this animal is we have produced an electron microscopy volume. This electron microscopy volume, which was produced for one individual, has been segmented. That means the computer has recognized every individual cell by its outline and also by its structures and can convey different colors. What you see here are the nuclei of the cells that have also been recognized by the computer. And also the chromatin that I showed you before. So this specific pattern in the nucleus is chromatin. So this animal, this one individual of Platinureus has been extremely well understood. It is part of this EM volume. And what we have also integrated into this atlas is gene expression. So this is conventional whole mode in situ hybridization, which has been mapped onto this atlas by registration. So we have mapped gene expression. For example, here is a muscle marker, which you can then see is expressed in muscles, and you have it in this, mapped on this EM volume. Yeah, I can show you in detail. This is how we can map gene expression here on EM, which means we have a species for which we know for all cells, both the gene expression and also structure of ultrastructure of individual cells and cell types for the entire body. So this is a rich research. I give you another example. This here, for example, is a part of the head. Here are the Platinureus eyes in volume EM. You see the pigment cup and also the lens. And what is shown in colors here are the genes, eye genes that are expressed in these cells. So a lot of information. We can use this information to cluster these cells. These are all the cells of the Platinureus body, clustered according to gene expression, as I showed you before. So for every Every dot is a cell, and the position of the dot is its position in gene expression space. This is based on something like 250 genes, and the cells that go together have a similar gene expression profile. If we map them back into our EM volume, you see that they very nicely match for example, the ganglia of the nervous system, the muscles in violet here, and different parts of the brain, and also peripheral ganglia, as you see them here. So we have a nice resource. Onto this resource, we can map the other information that we have is the connectomics information. This is illustrated here, and this is something where we have done something, but much more has been done by Gaspar's lab. Gaspar Jekli, also at COS, he's here in the audience. He has produced a lot of information on how the different neurons connect. This here is from our lab. We have only mapped a random set of neurons in the brain using this brain volume. Kevin in the lab has done it, so we know so these dots here represent neurons. The lines are axons that have been reconstructed from the EM volume, and you can see where they project. This is here from Gaspar's lab. This is this slightly younger young worm for which all the neurons and all cells in the body have actually been investigated in terms of connections. So all the neurons in the body have received their connectomics information. And that means our knowledge of the individual cells of the body is quite elaborate. And one novel characteristic that we have used these data for are the so-called morphofeatures. And for this, I would like to explain a little. So in this volume of electron microscopy, the computer has segmented cells. Yeah, we have the cells are these colorful dots here. These are individual cells in the body. It's not that many, but it's still 12,000. And the computer has segmented nuclei. It's these here. And for all of these nuclei, and for these cells, and also for the chromatin, there's always a shape that the computer knows. There's a certain texture context, so the computer can just integrate over the texture of these cells. And then what we also determined were so-called fine texture patches. So for each cell of the body, you can just randomly select little squares for which you determine the fine texture. And all of these, and then I think you will have heard about the new possibilities that we have with artificial intelligence, so-called deep learning. And we have used here a deep learning pipeline to ask the computer, in this space, we use this information, shape, texture context, and fine texture patches, so that in a way, every voxel in this volume gets a certain attribute, a certain color. And what we can then ask the computer is to find correlated voxels, correlations about voxels, and find out whether we can reduce this information into a lower dimensionality. What it does, you don't have to understand the whole pipeline here. What it does is that it comes up with a vector. This is a vector that has 250 variables in our case. And all of these variables get a certain numerical value. And this is the quintessence for what the computer tells us what a certain cell is like, because it's enough to have a value for these 250 parameters to reconstruct the entire cell ultrastructurally. And that is something that is typical for artificial intelligence. We cannot understand how the computer does it, but we know that these 250 so-called morphofeatures are enough to represent an entire cell. Because for every cell of the Platinere's body, there is now a so-called feature vector, and we have 250 variables. We have a numerical value for each, and if you give the computer the feature vector, it will give you the cell. And this takes advantage of all components, so shape of cells, texture of cells, fine texture of cells, and the same for nuclei. So also shape of the nuclei, the fine texture of nuclei, which is then the chromatin, and so on and so forth. And everything is part of these feature vectors. We also tried what components, shape, texture, and so on are most efficient actually in recognizing cells back. And I can tell you that, for example, the fine texture of the cells is extremely information rich in terms of telling cell types. So is the fine structure of the chromatin, which I explained you in the very beginning of my lecture. So the AI is able to extract this information, if you want, by a very complex correlation analysis, reduce it down in a low dimensionality space in the so-called feature vector. Yeah? So, and then gives you for each cell a vector, and this you can now use as morphological information for these cells and also compare it. And what we did here is, first of all, these are 250 variables. We can use these to put these cells into a dimensionality space with these 250 dimensions and project them there. And you see again that it looks a little bit like the UMAP from gene expression. You have the cells some cells coming very close together. See these here, for example, in the feature vector, they get very similar values. So they are, in feature vector space, they are very close together. And then when you look at it, what do they represent in our volume? They look very similar. They have similar shapes, they have similar fine structure. Yeah, so it is not by chance that the dots are so close here. And the same here for these ones. They are these cells, again, they are very similar, and so on. Yeah, so these are 3 examples of cells that the computer places very close together in the morphofeature space. And this way we recognize, okay, these are actually very similar cells. And we can then, of course, ask, so if we have here the cells plotted into morphofeature space, yeah, we can treat it like gene expression data. You have seen the UMAP about gene expression before. And we can then say, okay, we have a cluster here, cluster 14 is this one. Or we have cluster 11, these ones. And these are then morphofeature-based clusters of cells that obviously represent something interesting morphologically, but found by the computer in a completely unbiased manner. And for example, these 2 groups here, 10 and 11, represent 2 sets of morphologically related cells. One are epidermal sensory neurons, and we knew that they existed, yes, but this is a way of getting them all in the body, and these are the cells contained by this cluster. And the other one are epidermal cells, again, just recognized by morphofeatures. And so then we can compare, we can say, I was talking about the definition of cell types before. Now we have, I have 3 pictures here for the same volume. It's more or less the same region of the body. This is the brain. And these here are the manually segmented ganglia of the brain. And this here, for example, are the developing mushroom bodies in the Platinereus brain, this dark green area here. And so this was done by the postdoc working on this volume just by manual segmentation. He was going through the stacks and telling us, okay, here's a tissue boundary, and this is how I subdivide the brain into reasonable morphological units. Yeah? Then this here, on this side, is the subdivision of the brain based on expression profiling. I showed you that we have gene expression for the cells, and we can use this to also cluster these cells and put them into groups. And this here is the same cluster recognized by gene expression, and here you see the same mushroom body part of the brain recognized by morphofeatures. They are equally powerful. Yeah, so these morphofeatures that I just showed you, that we defined for the first time in an unbiased manner based on volume EM data, are in fact detecting these brain cells here as being morphologically similar by nucleus, by shape, by fine texture of the cytoplasm, and so on. And so where I would like your fantasy to go now is, okay, we have interesting perspectives here. We have cells in the body. For these cells, we can determine for example, by single cell sequencing, we can determine how they express each gene of the body, some of it very specifically. So we can produce a gene expression vector for each cell in the body by single cell sequencing, for example, with 10x. 10x machine is shown here. This is what we have in the lab. On the other hand, with volume EM, we can deduce morphofeatures. Yeah? There's a huge potential. We can do this so far we've done it for one individual. We can define morphofeatures also that are valuable, that are valid for the entire For a cross specimen, for example, we can define morphofeatures that are valid across species even, and then compare cells both at the level of these morphofeatures and also at the level of gene expression. So this is where we want to go in the future. We started with Platinereus, but now we want to produce similar data sets for other animals where we can get the entire body, both in terms of gene expression and also in terms of morphofeatures. And use this for now for the conservation of brains. So this was in a way outlining the tools that we are acquiring in the moment to do evolutionary comparisons, brain comparisons at a completely new level, yeah? And also unbiased. And so what I would like to combine this with is now single-cell transcriptomics for Platinereus, so gene expression and what we learned from it. This is now the whole body of Platinereus decomposed into individual cells and then sequenced. So this is a collection of 100,000 cells of Platinereus that have been sequenced. All of these people have been involved. And what you see is that, again, we get very defined little clusters. Some of them are bigger, and some of them are very small. So this is already the level of individual cell types. This is here a whole conglomerate of cell types, if you want. And we can, as you know, these are gene expression vectors conveyed to every cell. So you can use this information for similarity matrices and then for example, produce a neighbor joining tree. Here I would like to emphasize that this upper part of the neighbor joining tree has the neurons. But what you see already is that the neurons don't form one group. They form groups that are deeply subdivided. So this is here a group of sensory cells. This is a group of motor neurons and secretory cells that, okay, they come from the same root here, but the support is low, and they form two deeply subdivided groups. So we can use this tree, and put the cell types into clades, so-called clades, so groups of related cell types. And these are these 11, I think 14 clades altogether that we deduced from our single cell information. And the neurons are here. This is the biggest clade 11, clade of neurons. Then these are the mushroom bodies of the animal, and here are the sensory neurons of the animal. And I'll talk a little bit about this. Just a few words on these sensory cells, on the mechanosensory girdle. It has if you look into transcription factors that define them, you come out with the same transcription factors that are specific for this entire group, like POW4 and FOXJ, as, for example, you have for the sensory neurons also in the chiton. And you can then ask, what are the most specific genes for all of the sensory neurons in the body? You can come out with this list, which is, again, very similar to other organisms. And you can also ask, okay, what are these cells that belong to the sensory girdle? We can do this because we have the EM volume. These are the antenna, the palpi, and the peristomial cirri of the body. And also the sensory cells of the periphery. So here are two of the marker genes and their expression. And you see that they are in the sensory organs around the body in our annelid. So we can directly do the morphological analysis. Here are some of the sensory cells in the lateral appendages. And if we then go into detail, taking our EM volumes, we can find these cells back. This is the cell expressing brain 3, for example, the marker transcription factor. And it is a sensory neurons that is, in fact, resembling a hair cell as we have them in our inner ear with a hair cell-like collar. And this is a picture from Gaspar's lab, who has found the exact same group of sensory cells by connectomics. So this is a slightly earlier stage. Here are the sensory cells in the periphery and also in the head. And this is how they project into a girdle of axons that surrounds the midline of the animal, and he has termed this mechanosensory girdle. So we see this in connectomics and also, for example, by gene expression in single-cell data. How about this clade 11? Clade 11 is the biggest clade of neurons, and I just use it to illustrate you the complexity of this data set. What you see is that we just took the cells of clade 11 and subclustered them, and now you see the cell types emerging. So these are probably cell types that at least count in several dozens, if not 50 to 100 cell types that are neuronal cell types of the ventral nerve cord and of the brain. And each get their different numbers here. I won't go into any detail, but for example, if you take this cell type 49 and ask for the genes that are specific for this cell type, the coregulon, this is interesting because this is a cell type expressing R-opsin 3, which is one of the rhabdomeric opsins of the animal. So this is obviously a photoreceptor of some kind. And you see that this R-opsin is pretty specific for the cell type where it shows strong expression. So this is for sure real. We have another cluster, cluster 4, which lies here. And cluster 4 has been subclustered as well. And this is the cluster having two cell types that express the ciliary opsin in Platinureus. So this is the opsin that we share with worms that is expressed in our rods and cones. So these are photoreceptors. And then we can ask our resource, what is this? If we have rhabdomeric and cells expressing rhabdomeric opsins and ciliary opsins, what is this morphologically? And this way we discovered the templin eye, which was also already found by manual inspection of the 6-day volume by Rachel Templin. It was an eye that we were completely unaware of, which exists in the animal. She went into much ultrastructural detail, and all that I want to tell you here is that it is a very weird eye where two types of photoreceptors, ciliary and photoreceptors, go into the same pigment cup, which is this pigment cup here. They all bring their photosensitive cellular protrusions, both ciliary and rhabdomeric, into this cup, and then this eye prevails even into late juvenile stages. So this is how far we could track it. And this is quite intriguing because we have rhabdomeric and ciliary photoreceptors together. I put this in actually for Jochen. He's unfortunately not here today, but this is for something who is interested from the perspective of photoreceptor evolution. This is something very interesting in terms of co-presence of ciliary and rhabdomeric photoreceptors in one eye, and we were completely unaware of this. So this we can recognize morphologically, we can recognize in terms of single cell data, and now the last minutes I would like to talk about this clade here, the mushroom bodies. So these are neurons that form a distinct clade, again deeply separated from other neurons, and they form the cortical structures of the animal. This some of you might remember from yesterday's presentation. We are very interested in these mushroom bodies, because they are the brain structures that are morphologically known also from insects, mushroom bodies. In insects, it's well established that they are the sensory associative centers of the brain. So they are, in a way, the learning centers, and by morphology have always been assumed to be something similar also in annelids. And now we wanted to go into a morphological detail and expression detail for these mushroom bodies. Subclustering of clade 9 brings us a smaller number of cell types. I will focus on this cell type 48 because it really illustrates well the power that we have from single cell data. We have a lot of cell type specific genes and transcription factors like these ones that are specific for the cell type. So this is a small set of factors that is obviously involved in probably the specification of mushroom bodies in Platinureus. These are interesting genes because many of them have been known before to play a role, conserved roles in chemosensation. In Drosophila, like IRX, aristales, it is needed for arista formation. It specifies a subset of chemosensory neurons in the nematode. It is essential for development of mouse olfactory system. And PTF is FAIR1, is one of 7 transcription factors required for chemosensory neuron specification in flies. I won't go into detail here, but these are not transcription factors so they tell us a story actually that these mushroom bodies seem to have a link to chemosensation. And now looking into, maybe I'll first go here, into what these mushroom bodies really are, is that if we take the 2 specific transcription factors, ARX and PTF1, and plot them in our Plati browser, we get we indeed come here to the calyx of the mushroom body in Platinereus. This is the only site where these two transcription factors are co-expressed. This is probably more still the proliferating cells where ARX is already on. And then you have these cells here that belong, are part of the distal calyx of this brain structure in Platinereus. And we looked into detail again, and what we found for these cells is that it is a mix of interneurons. Interneurons are in red. But what we also found as part of the mushroom bodies is that the cells have sensory appendages that reach the cuticle. So this comes from tracing the sensory endings. And if you reconstruct them in EM, you see that these are, they have cilia that run underneath the cuticle via canals. They are connected to the outside. So these are very likely chemosensory neurons. And it makes a lot of sense that both by expression and also by morphology, we identify a direct sensory component for these mushroom bodies in Platinereus. So if you, at the same time, these sensory cells project into the neurosecretory plexus, which we identify as a homologue of the neurohypophysis in vertebrates. And so this is interesting because we can assign a chemosensory characteristic to these mushroom bodies in the annelid worm, which was completely unknown of before. But it is interesting because we are dealing with the putative sensory associative structures, which would be in our brain, the cortex, and in the fly brain, also the mushroom bodies. And it has always been known that there is some strange link between, for example, our cortex and chemosensation, which is the direct connection of our telencephalon, our cortex, to the nose. And what we postulate now is that the sensory associative structures in animals have evolved or started the evolution as chemosensory organs proper, and then specialized on the associative aspect of their function. And so this is the data that this hypothesis is based on. And then we can try to compare these structures across animals. How do we do this? This is this UMAP that I show you here, where we have integrated cell type expression information for lamprey vertebrate, for Amphioxus, annelid, and ant and fly. And these are brains. So that we took from published data sets. And then you can ask, okay, how can you compare expression information between, for example, a lamprey and an annelid and put it into the same expression space? Well, what we did for this is we reconstructed the genome of Urbilateria, and that we didn't map expression information on specific genes of the genome, but on orthogroups of related genes that we identify in all bilaterians. They are called orthogroups, and they correspond to the Urbilaterian genome. Yeah? So we can theoretically reconstruct the Urbilaterian genome because these are genes that exist in all the big bilaterian groups, and map it onto those, and this way then directly compare expression, for example, between a lamprey and a fly. And if we do that and ask where are the mushroom bodies here, then the annelid mushroom bodies, the cells from the annelid, they come to lie here. So this is where they come to lie in the shared expression space of all these species. If you then look where the ant mushroom bodies, the Kinyan cells of the ant mushroom bodies come to lie, it is here, which is this. Many of these are overlapping in the same region. They also populate other spaces, like here, where there are also some annelid cells, actually. So this is a good hit in terms of cells from the ant mushroom body, so hundreds of millions of years in evolution away, and annelids going to the same region. And what is interesting is that the fly mushroom bodies are kind of in the neighborhood, but they are not exactly matching. So there's more of a similarity in expression profile between ant and annelid than there is between ant and annelid and either of the two and fly. But still it's related. It's clear that ant and fly mushroom bodies are evolutionarily related. So this is giving you a glimpse of what is possible now. So we can take this expression information that we have for different organs, for different cell types in very different species, and see where they end up in expression space between very remotely related groups only. Okay, and where do we go with this? Well, we are in the moment reconstructing, bringing all the information together, trying to reconstruct everything. And for the annelid, this means we have identified some fundamental units, such as the mechanosensory girdle. The components are depicted here in yellow. Motor girdle, I didn't talk about this, but these are motor neurons that are located around the neuropil that is along the midline of the body and around the foregut. And on top of this, we also have sensory associative centers that are in blue here as major components of the brain, and also an apical nervous system. I didn't talk about it, but these are cells that would correspond to hypothalamus in vertebrates that are neurosecretory cells of the brain. So these are the elements, the elementary units of the annelid brain. And what I would like to close with, what is possible so far is that we can compare these elementary parts to parts of the developing vertebrate nervous system. So all of these components, the mechanosensory girdle, motor girdle, sensory associative centers, apical nervous systems, are components that you also find in the vertebrate brain. The mechanosensory girdle by transcription factor identity would correspond to the lateral parts of the neural tube here in yellow. Then everything that is beyond the zona limitans, this is the alar plate. Then you have the basal plate of the neural tube, which is the motor girdle in annelids. You have the hypothalamus here, which is in violet, which resembles the apical nervous system. And then you have the telencephalon, the cortex, the developing cortex, which would be the sensory associative centers of the vertebrate. So we are trying to establish one-to-one correspondences between brain parts in annelids, in vertebrates. We are reaching out into other organisms, mostly in collaborations with other labs, doing single-cell sequencing, doing volume EM, and this way then try to put these comparisons onto larger and larger grounds. And I've mentioned many people on the way doing this work. This is the lab from last year. This year's lab retreat will only happen later this year. And yeah, we have a network. It used to be EvoCell, now it is ZooCell, where we bring together a lot of labs working with a similar approach on comparing cell types, both gene expression-wise and also EM volume-wise, across phylogeny, trying to reconstruct the evolution of many organs actually, but with a strong focus on nervous system. And yeah, this is maybe interesting for you. Many of these labs, including ourselves, are also welcoming pre- and postdocs on this topic to, in a way, revolutionize evolutionary comparison with modern techniques that so far had not been used in this entire field. And what I would also like to mention and to close is that this is an extremely data-rich discipline. Evolution has always been an extremely data-rich discipline because you need to know about a lot of animals, a lot of organisms, if you really want to come to reasonable comparisons. This is now in so much detail with, for example, imaging data and expression data that these whole comparisons will be, I think, a really exciting data field of data sets for artificial intelligence, obviously, and for deep learning. I gave you some glimpse of it today, but it is very obvious that if we do it right, many of these evolutionary questions will probably be solved by means of AI. And maybe this is also to stimulate some discussion and some thinking. It could be one of the most exciting paradigms, actually, where these new developments can be extremely fruitful. And I think I'll leave it with this and take questions.